everyone. This is the part of the specimen collection and transport media series. We're talking about culture swabs. And these are also known as culturettes, double swabs, aerobic swabs, and really what they're called depends on the lab in which you work and the manufacturers too. There are two main manufacturers of these kinds of swabs. This one is manufactured by BBL, which is a Becton Dickinson company. For this video, I'm going to explain what these swabs are made of and how they work with the bacteria. And then the last part of the video, I'm going to show you how to use them correctly while collecting and transporting your specimens. This swab here is manufactured by Copan, but you can see from the last slide, they look almost exactly alike. And they're almost exactly alike as well. So you can see that there are two swabs here. We use both sterile swabs to inoculate our media plates, and then we take one swab out and we make the gram stain. And since the slides that we use for gram stains are not sterile, we discard that swab, and then we still have one sterile swab left over to keep in case we need it. Also, you can use one of these swabs for PCR testing and then use one swab for culture. You can do that. The swab tips are made of rayon. Some materials like cotton, contain compounds like cotton contains fatty acids that are actually toxic to some bacteria. So a synthetic non-toxic material is used for the swabs, and then the shaft is made of plastic, another, non-reactive material. The foam stuff here on the bottom is saturated with liquid amines. This is a preservative that is a non-nutritive preservative, but it does preserve the bacterial viability, so it keeps them alive, even the most fastidious bacteria. It also prevents bacterial replication of the organisms, and that's necessary if you want to see how much bacteria was in your actual specimen. And when you think about that, some bacteria can replicate every 20 to 30 minutes. If you don't have something that stops the replication within a couple hours, your bacterial count is going to be so much larger than what was actually there in your patient specimen. So you need some sort of preservatives that stops bacterial replication so you can quantify the actual amount of bacteria that's in your patient specimen. So this is not for obligate anaerobic organisms. This is not an oxygen-free environment in here. So if your provider asks for an anaerobic culture, this is not the swab you want. Okay. Always check your expiration date, which is found up here on the upper neck here. And also, see this foam stuff down here? If you see it's all dried out and crusty, don't use the swab. There's a good chance that it could be expired because after a while they dry out. But the preservative is called liquid amines for a reason, and it's not going to work if it's all dried out. And then if we can't recover the bacteria, then we're going to have to recollect. Patient care is adversely affected. It might delay treatment. So always just make sure you pay attention to the expiration date, and make sure this isn't all dried up and crispy. Okay, so to recap, this is for bacteria that can grow in the presence of oxygen and basically keeps the bacteria alive but doesn't allow them to replicate. Okay, so for the next part, I'm going to actually show you collection and things like that. All right, thanks. So you can see here, this is a Copan swab, and it's supposed to be completely sealed. I accidentally opened it, so just pretend that it's sealed. First thing you're going to do is you're going to open it aseptically. Everything on the inside of this is sterile. Grab the white tube. Once you grab the white tube, the outside of the white tube is no longer sterile, but the inside is. You want to check to see the foam there on the bottom that has the liquid amines in it. You want to check your expiration date, and you also want to label your specimen with 2 patient identifiers or a patient label. So you can kind of crack the white lid there because everything on the inside is sterile. You want to maybe keep the lid on there until you're ready to put your swabs to maintain sterility on the inside of your tube. So now you're going to take the 2 swabs. You can see that there's 2, and you can remove them from the lid, but you really shouldn't. You shouldn't have to. So leave them in and make sure they're in there firmly, because we don't want them to fall out inside the tube, because we can't get them out while maintaining asepsis. Another thing you'll notice is there's some score marks here on the end of the shaft. Those are for us in microbiology, because sometimes we have to incubate the tips of the swabs because the shafts will not fit in the tubes that we need. As you can see here, the shafts will not fit in this tube. So what the score marks are for us in micro, so we can snap the tips off into the liquid. You will never have to do this, and so you should never ever snap those tips off. When you put it back in the swab, or when you put it back in the tube, the swab should be seated firmly in the lid and should all be in one piece and not snapped. Okay, so what you want to do then is when you are taking a specimen, you want to make sure you kind of roll it around and saturate both swabs. You also want to make sure that you just stick to where the wound is. You don't want to like smear all around where the skin is too, because the skin will never be sterile. Okay, so you're always going to get some normal microbiota, so stay well within the wound. All right, so then you're going to take the white cap off. You can get rid of that. Gently put your swabs in, tighten the cap. All right. And you can get rid of the white cap then, because that was just a holder. And as long as make sure you got your label on it, you're good to go. All right. Thank you.